I'm very honored to present the David Marsden lecture, which will be given by Professor Hiroshi Shibasaki. The learning objective of this talk is that at the end, participants should be better able to understand the physiological mechanism of cortical myoclonus. Professor Shibasaki was born in the Hyogo Prefecture, Japan. He received his MD and PhD from the Kyushu University, which is located in the beautiful seaside city of Fukuoka. This was followed by training as a resident of neurology at the University of Minnesota Hospital in the US. Afterwards, he returned to Japan and he became affiliated to the Kyoto University School of Medicine, where he has developed most of his career. At that institution, he became professor and later chairman of neurology. He's currently professor emeritus of neurology in Kyoto. I'd like to highlight that in addition to Professor Shibasaki's scientific contributions, and we'll go back to that soon, he has had a continuous international activity, including as president of the International Federation of Clinical Neurophysiology. Thanks to his very active networking and connections with international colleagues, he has been instrumental in inserting the Japanese neurology in the international scenario. Now moving to his connection with Professor David Marsden, it's interesting that one of his spells abroad was at the Institute of Neurology, Queen's Square, London. I'm sure that you have a chance to address that in his talk. And with David Marsden, he has shared an interest in clinical neurophysiology and myoclonus. So I believe that it is very fitting that the organizing committee of the MDS virtual invited him very deservedly to present the C. David Marsden lecture. Professor Shibasaki, please come to the podium to give your talk. Thank you very much.
It is my great pleasure and a great honor to give this David Marsden Award lecture. I'm not sure if you can see my cursor moving, but this photograph was taken, I believe, in Washington, D.C., 1990. And I looked like this 30 years ago, and so did, from the right to the left, Joe Jankovic, Warren Olanoff, C. David Marsden, Alfred Bernardelli, and Bastian Conrad, and Mark Hallett. My interest in myoclonus dates back to 1971, when I was a resident of neurology at University of Minnesota Hospitals in Twin Cities, United States. I was recording sleep EEG in one of the patients with myoclonus, and in that patient, reflex myoclonus takes place only during REM sleep. And that's what I found, and I reported the case in neurology. The department chairman at that time was Professor A.B. Baker. He was really an excellent teacher of clinical neurology, and in fact, he was a founder of American Academy of Neurology. I wanted to complete my residency there, but my boss in Japan was worried that if I completed residency, I might stay in the United States without returning to Japan. So I did come back to Japan, and I kept my interest in myoclonus. I'm not sure if you are able to see this video as a moving picture. But this patient has progressive myoclonus epilepsy showing postural as well as action myoclonus. And one day, I was trying to record Bragg shaft potential in this patient. Bragg shaft potential is called readiness potential, which is a slow electroencephalographic activity starting 1 to 2 seconds before the voluntary muscle contraction. Since this activity is very small, we have to average EEG with respect to the voluntary muscle contraction. So I was expecting to see slow EEG activity, if any, but I happened to notice a very strange sharp activity on the EEG, time locked to this EMG activity. Looking back at the original record, without averaging, there was nothing particular on the EEG, but there were a lot of frequent small discharges in the EMG corresponding to myoclonus. So here, the computer averaging system was triggered by myoclonus instead of voluntary muscle contraction. And so this activity was seen preceding myoclonus of the hand by 20 milliseconds and distributed over the central area contralateral to the myoclonus. Later, I worked in Queen Square, London, actually in Martin Halliday's lab. And Dr. Halliday names this technique jerk-locked back averaging. For this technique, we record EEG and EMG simultaneously, and by using the EMG of myoclonus as a trigger, and back average the EEG. This is actually moving picture, but I'm not sure if you can see it as a moving picture or not. The patient showing action myoclonus. What is most important for this technique is to find out the best muscle for recording myoclonus. Actually, we can average EEG while we are actually acquiring data of EEG and EMG. But we come across with very noisy record because of a lot of background EMG activity triggering the averaging system. Again, I'm not sure if you can see this video as a moving picture or not, but it is useful to do offline averaging. In other words, we save all the data in a digital form. We look at each individual EMG to find out the most appropriate, best muscles, best EMG activity associated with myoclonus. So we only choose the myoclonus activity, which is subjected to trigger the averaging system. Here you see EEG activity standing out time locked to this myoclonus as the number of sweeps increases. So physiological study of movement disorders, it is important to do it by yourself. I could have easily missed this serendipitous observation if I was not doing it by myself. This is an example of polygraphic record of EEG and EMG from another patient with progressive myoclonus epilepsy. We see a lot of frequent sharp discharges in the EMG corresponding to myoclonus. And we often see spikes on the EEG in association with myoclonus. However, on the conventional polygraphic record, it is difficult to study the relationship between spikes and myoclonus precisely. Therefore, we can use this myoclonus as a trigger to back average EEG. This is the average record of EEG, time locked to myoclonus of the right hand in this case. And this activity here precedes myoclonus of the hand by 20 milliseconds and distributed over the central area, contralateral to the hand from which myoclonus was recorded. If magnetoencephalogram MEG is available, we can obtain the generator source of spike with better spatial resolution. Here, MEG was averaged with respect to the myoclonus of the right hand. And here the small spike, very localized to the central area, contralateral to the myoclonus. Based on the MEG data, we can calculate the generator source of spikes. This is estimated source of cortical spike preceding myoclonus of the right hand in 3 different cases with cortical myoclonus. As you see here, the generator source of the spike is localized to the hand area of the precentral gyrus. As you are aware of, cortical myoclonus is often stimulus sensitive. I'm not sure if you can see this video as a moving picture or not. Now, tapping the left elbow causes positive myoclonus in the right hand, in the left hand, I mean, and then which is followed by another jerk in the right hand. I will repeat. Tapping the left elbow causes myoclonus in the left elbow, left hand, which is followed by another myoclonus in the right hand. And this is actually what is happening. Here, the left hand is electrically stimulated, and giant HCP is recorded from the right central area, which is followed by reflex myoclonus, or C reflex in the left hand, which was stimulated. And 10 milliseconds later, there is giant SAP also recorded from the left central region, which is ipsilateral to the stimulated hand. That is followed by another reflex myoclonus in the right hand, which was not stimulated. The time lag is 10 milliseconds. And this 10 millisecond time lag corresponds to the time interval needed for impulse transmission conduction through the corpus callosum. Now, in the sensory motor cortex, this is what is actually happening in the cortical reflex myoclonus. This is somatosensory evoked electrocorticogram in a patient with medically intractable epilepsy starting from the left foot. This is a part of a pre-surgical evaluation. And here, left foot is electrically stimulated, and the giant ACP is seen in the foot sensory area, and 5 milliseconds later, that is seen in the foot motor area. So this is actually what is taking place in the sensory motor cortex in the cortical reflex myoclonus. This schematic diagram illustrates the possible mechanism underlying generation of cortical myoclonus. In the spontaneous cortical myoclonus, the spike is generated in the primary motor cortex, and the impulse is conducted through the corticospinal tract and alpha motor fibers of the peripheral nerve to produce myoclonus in the corresponding hand 20 milliseconds after the spike. In the cortical reflex myoclonus, the somatosensory cortex is also hyperexcitable. Therefore, incoming stimulus produces giant ACP, and through the reflex mechanism in the sensory motor cortex, the impulse is conducted through the corticospinal tract to produce reflex myoclonus, which is also called C reflex. And so here, 20 milliseconds for the afferent pathway and 20 milliseconds for the efferent pathway, which produces 40 milliseconds after the stimulus. And as you know, EMG discharge associated with cortical myoclonus is very short. And that is because the impulse is conducted through the fast conducting fibers of the corticospinal tract without temporal dispersion. Now then, how are the muscles involved by cortical myoclonus? Now here, the myoclonus recorded from various muscles arranged from the proximal to the distal muscle of upper extremity. I would like to draw your attention to call your attention to this pair of muscles, which is with extensor and flexor. So agonist and antagonist muscles. Please notice that these two muscles are involved by myoclonus nearly at the same time, both for spontaneous myoclonus and for evoked myoclonus. So the primary motor cortex is muscle-oriented, but not movement-oriented, because regardless of whether spontaneous or stimulus-induced, cortical myoclonus involves agonist and antagonist muscles nearly at the same time. If movement-oriented, it is expected to see the reciprocal innervation. Now here we are looking at the spread of myoclonus from proximal to the distal muscle, both for the spontaneous myoclonus and reflex myoclonus. The myoclonus spreads from the proximal to the distal muscles rapidly. So the impulse conduction through the corpus callosum causes 10 millisecond delay of myoclonus in the opposite hand, because the time interval needed for the impulse conduction is 10 milliseconds. Then the impulse transmission within the primary motor cortex causes rapid spread of myoclonus to the adjacent muscles. I'm not sure if you can see my cursor moving or not. So this is indicated by A for the transcallosal conduction and B for intrahemispheric impulse conduction transmission. Then I'm going to talk about positive motor phenomena and negative motor phenomena. Again, this is a moving picture, but I'm not sure if you can see it or not. This patient with progressive myoclonus epilepsy has frequent jerks, shock-like movements due to abrupt muscle contraction. But we often see big drop of arms, which are due to interruption of muscle discharge, negative myoclonus. And this is the EMG trace from a single muscle from the patient that you have just seen on video. So here, abrupt instantaneous increase in the EMG discharge is positive myoclonus. This is conventional myoclonus. Here, the sudden interruption of the ongoing muscle discharge causes drop of the arm or negative myoclonus. We often encounter a combination of the two, positive and negative myoclonus. Now, cortical negative myoclonus is also stimulus sensitive, just like cortical positive myoclonus. Again, I'm not sure you can see this as a moving picture or not. The tapping of the left hand causes hand drop in the left, and also in the other hand with short time lag. These are polygraphic records from another patient with cortical reflex negative myoclonus. Here, the left hand is electrically stimulated once every second, and each stimulus produces giant ECPs as indicated by white arrowheads. Each individual stimulus causes giant ECP, and since this is so large, we don't need any averaging. And then this spike is followed by interruption of EMG activity, which is silent period, corresponding to negative myoclonus. Therefore, this is cortical reflex negative myoclonus. The next slide is going to show you one of these events in a magnified view. Here, the left hand is stimulated, giant ACP over the right central area, followed by the silent period, the stimulated left hand. Another giant ACP over the left central area, which is ipsilateral to the stimulus, which is followed by silent period in the right wrist extensor, which is not stimulated. So here we are looking at exactly the same finding as the one that we saw for positive myoclonus. Then what is taking place in the primary motor cortex for these negative phenomena? It is postulated that there are two different components in the primary sensory motor cortex. One is excitatory and the other is inhibitory. When the excitatory components are either stimulated or involved by epileptic spike, it causes positive phenomena such as positive myoclonus. When the inhibitory components are either stimulated or involved by epileptic spike, that produces negative phenomena such as negative myoclonus. And it was it was a case, in fact, and we had a chance to record electrocorticogram from across the central sulcus in a patient with medically intractable epilepsy as a part of pre-surgical evaluation. So here, subdural electrodes are placed across the right central sulcus. And when this pair of electrodes is stimulated by a single pulse, we see positive motor phenomenon, muscle evoked potential in the corresponding muscle, followed by silent period. And when stimulus intensity is increased, the amplitude of this muscle evoked potential increases. But when this pair of electrodes is stimulated, it produces only the silent period in the ongoing muscle discharge of the contralateral hand. And there was no preceding positive motor phenomena. When the stimulus intensity is increased, the silent period becomes longer. And the longer silent period is associated with clear negative myoclonus or asterixis. Dr. Hans Luders of Cleveland proposed the presence of negative motor area in both primary and supplementary motor area. And what we saw in the myoclonus patient corresponded to negative motor areas in these areas. So negative motor phenomena are caused by either spontaneous or stimulus-induced activation of the negative motor area of the sensory motor cortex through an active process at the cortical level. I say here active or excitatory because it is preceded by an epileptic spike. So in conclusion, cortical myoclonus is caused by spontaneous or stimulus-induced epileptic overexcitation of the primary motor cortex, and by the fast impulse conduction through the corticospinal tract. Then the impulse transmission within the primary motor cortex causes rapid spread of myoclonus to the adjacent muscles, and the transcallosal impulse conduction causes a 10 millisecond delay in the opposite hand. There are negative motor areas in primary motor cortex, which cause negative myoclonus when stimulated or involved by an epileptic spike. So in cortical myoclonus, we are looking at the overexcited behavior of the physiologically existing central motor control system in humans. Thank you very much.